So we can move on to a talk by Luis Palomira Bueno on the identification of pathogenicity factors and first detection of Xylella in Colombia. And Luis is a postgraduate student at University of Reading, and she's based at NIAB in Cambridge, and she's funded by AHDB. Her work focuses on Xylella virulence, and she also undertook some sampling in Colombia, and she's going to tell us all about it now. Over to you, Louise.
Can you all see my slides?
Yes, that's perfect.
Okay, perfect. Thanks, Joanna, for the introduction. So, because I only have 10 minutes, I will be mainly presenting our work that led to the first detection of Xylella in coffee in Colombia. But if you have any other questions, just contact me. And yeah. So first of all, I would like to say that this project only happened with the help of all these people. I want to thank the AHDB for funding this project and my industry partners, Brian and Jamie, my supervisors, Michelle, Emma, Richard, Rob, and Glyn, and most of all, our Colombian collaborator, Valeska. Thank you all for the help. So this diagram shows where Xylella has been detected globally. We know that Xylella fastidiosa is very common in the Americas, but also in Europe and the Middle East. And in Taiwan, the species Xylella taiwanensis has also been reported. So why did we decide to go sampling in Colombia? Xylella has been found in many surrounding countries of Colombia. So we've got Costa Rica, where Xylella has been found in coffee, citrus, avocado, and oleander. Puerto Rico, where Xylella has been found in coffee and citrus. Venezuela in grapevine. Brazil in citrus, coffee, and plum. Paraguay in plum, and Argentina in citrus and olive. And Xylella hadn't been reported in Colombia, even though it is one of the most biodiverse countries in the world. And also Colombia is a large coffee producer, and all 4 subspecies of Xylella that are common in Europe, so Xylella fastidiosa, multiplex, sandii, and pauca, have been detected in coffee. We visited several sampling locations in the Antioquia department of Colombia, which is here. These included a coffee farm, a citrus farm, 2 research stations, a rainforest, a university campus, and a location in a rainforest, sorry, a university campus and a botanical garden. And in total, we collected leaves from 51 plants, of 16 different species from 3 families. And we collected both healthy and disease-looking leaves. So these are pictures of coffee leaves. And this is a list of the plant species that we sampled. The green highlighted species are plants where Xylella had previously been detected in. And we sampled a lot of coffee relatives, the Rubiaceae plant species here, because for the 4 subspecies commonly found in Europe have been found in coffee. So we wanted to look at the relatives of coffee. Now I'll talk you through how we completed the sampling. So let's say we are at a coffee farm, and this is a tree of a particular cultivar. From this tree, we chose 3 branches. And from each branch, we collected 3 or more leaves. And we did this with another 2 trees from the same cultivar, meaning that we could perform 3 DNA extractions from each tree, and that we would have triplicates of each sampled species. So the leaves were processed first by surface sterilization. Each leaf was cleaned with 70% ethanol, followed by distilled water. Then we cut up the leaves so that we were only left with the midrib, basal, and petiole. Midrib, basal part, and petiole. And these would be homogenized in liquid nitrogen with a pestle and mortar. And then to extract the DNA, we followed the EPO protocol to detect Xylella in the leaves. So as primary detection method, we used PCR to detect the Xylella-specific RPOD gene, which codes for RNA polymerase sigma factor. And if this tested positive as verification, we ran another PCR targeting a different region in the genome, this time the 16S23S intergenic spacer. And then for some samples, we performed multi-locus sequence typing, which is a way to characterize bacterial isolates by looking at certain loci in housekeeping genes. And in the protocol that I used by Yuan et al., this is done by targeting 7 housekeeping genes. From the total of 51 plants that we sampled, we detected 13 to be positive for Xylella. These included 5 cultivars from one coffee farm. So the cultivars Geisha, Colombia, Catura, Pajarito, and Castillo. The A signifies an asymptomatic sample, and S signifies a symptomatic sample. So we've collected both healthy and diseased-looking plant leaves. And then a 13th sample that detected positive for Xylella originated from a university campus, of which the cultivar is unknown. And this is a gel that shows all the samples that tested positive for the Xylella-specific RPOD gene, so the first round of detection. The amplified sequence is 733 base pairs long. And as negative control, we use distilled water and coffee DNA only. And as positive control, we used spiked coffee DNA with spiked Xylella, and also Xylella DNA, which was Xylella fastidiosa, temecula 1. So from the 13 positive samples, we performed MLST on 4 of the samples. This allowed us to confirm the subspecies Pauca in all 4 strains. So what does this mean for Colombia now? Because no serious economic damage has been reported, this detection will not affect Colombian coffee production at all, which is actually the case with Puerto Rico at the moment. Colombia doesn't have the same regulations as the EU, so there is no need to destroy everything. But for us Xylella researchers, it would be very interesting to genome sequence the Colombian strains and see how they differ to the strains found in Europe. These results are currently in the progress of being published. So to summarize, we are reporting the first detection of Xylella in Colombia in 13 coffee plants from 5 different cultivars and 1 unknown cultivar. And we were able to confirm the subspecies Pauca in 4 of the strains. Thank you, everyone, for listening, and I'm happy to take any questions.
Thank you very much, Louise. That was really good. I can't see questions in the chat, so I'll ask one. Are you in contact with any researchers in Colombia? Could you collaborate with them to do more sampling? Well, for you, it might not be so necessary, but for them, it would be really interesting to find out and to do more sampling there.
Yeah, so I am in touch with our collaborator, Valeska, who practically organized the sampling in the coffee farm. And we haven't talked about more sampling opportunities yet, because we're trying to get the paper out first. But it would definitely be interesting to do more sampling, especially in the southern region of Colombia, to see if there's also Xylella over there.
Yeah, so we have a question from Elvesio. Do you have a sequence type for the Pauca?
Sorry, I should have put that on the slide. Yes, I do. So let me see. Okay, so for the two geisha cultivars, it was sequence type 66 and another sequence type 74. And from one cultivar, Colombia, it was sequence type 73. And the unknown cultivar was sequence type 74. So I used the EPO Xylella protocol, the very big document that summarizes all the Xylella research to see which sequence type refers to which subspecies.
So you have some diversity there straight away in Pauca. Another question from Robert Taylor. Did you manage to culture Xylella from these plants?
I have not tried that. I was going to, but then COVID happened actually. But no, we didn't try to culture it at all. All we did was trying to detect DNA from the leaves, from the total leaf DNA extracts.
And could it be difficult because the samples are frozen?
Yeah, so the sampling, we did this in 2019, and the samples are all in -80, so I'm not quite sure if the bacteria have survived. I haven't actually tried anything, but if anyone knows the survival rate of Xylella, be great if they could help me out.
And just one more question. To have the whole genome sequences, what would you need? Would you have enough DNA, enough material for that or not?
We would have enough DNA. We were thinking of doing whole genome sequencing using the nanopore, but that's been on hold because of COVID as well. But we would definitely have enough DNA to do whole genome sequencing.